Hello everyone, thank you for stopping by. My name is Cesar Tawfeeq from California State University Northridge. I will be introducing our IgStRAND numbering scheme for the immunoglobulin fold, which stands for IgStRAND residue anchor dependent numbering scheme. So this was done with the help of the NCBI and the NIH. And at the beginning, I'm going to go through a little bit of an introduction. So Ig domains are highly ubiquitous in nature and make up about 2% of the human genome, which is a large percentage. They also make up the structures of antibodies, like you can see here on the right. They're also present in a variety of protein types utilized for a variety of cellular functions, both as soluble and membrane-bound form. So the structure of an Ig domain essentially is made up of at least 7 anti-parallel beta strands arranged as two sheets, as you can see here on the bottom. And they have actually highly conserved tryptophan on strand C. They also have a highly conserved disulfide bond between the B and F strand on the opposite sheets. There's another highly conserved residue on the F strand, which is a tyrosine as well. But more important than these, there are actually highly conserved patterns of hydrophobic residues distributed throughout the Ig fold, which are essential for the formation of the Ig domain. There are actually different types of Ig domains, which are folded somewhat uniquely. Some can have an extra strand or a deletion of a strand, but despite their differences, they actually share common features, which put them all under the same Ig domain umbrella. So interestingly, the bends of the beta strands and the variable lengths of the loops, those two features allow for infinite diversity in the structure of Ig domains, which allow for high affinity binding and selective targeting of infinite number of ligands like organic molecules, proteins, DNA, etc. However, one thing actually slowing down the development in the science of Ig domains is the lack of a universal numbering scheme, which we can apply to all Ig-like folds and not just the antibodies, especially those involved in the immune system and in the nervous system. So we used in-depth structural analysis, we used structure-based sequence alignment, MD simulations, and also the conserved domain knowledge from the CDD in order to guide us towards choosing our anchoring and analyzing these different Ig domains which are listed here. So to introduce our numbering scheme, it is made up of 4 digits. The first digit here is for the strand number. So if it's an A, it's a 1. If it's a B, it's a 2. If it's a C, it's a 3. And if it's a 4, if it's a D, it's a 4, etc. And the second character here actually takes into consideration the presence of the prime or double prime. So for the A, since there is no prime, we just give it as a 0. However, when there is a prime, it's a 2. And when there is a double prime, it's a 7. And you can see that here both in the C and in the A, and also the G has a prime as well. And in orange, I show the anchor residues both in the structure and the sequence as well. And this example is from IgV of CD8 alpha from 2-ATP. Using the same molecule, we analyzed the rest of the sequence and we assigned the anchor residues for each strand, and we also aligned it so that it's easier to see. And you can see that, for example, if we take the F strand and we know that there is a cysteine here, the most important position, which we give it a number of 6050, and with that we can use it as a reference to number the rest of the residues on that strand. For example, we mentioned earlier there's a tyrosine, and this is the 6048 number for the tyrosine. And so all Ig cores, even though they can have a deletion in one of the strands, they actually do have a core. So the core is essentially the B, C, and E and F. So the B, C, E, and F. And here we have it also align structure-based sequence alignment of all the Ig domains that we analyzed. And you can see, for example, in the B strand, where there is usually a cysteine, but in some cases like the fibronectin 3 and the C2 of the CD4, we have a change. We don't have a cysteine there, we have different residues. And similarly in the C strand. So despite lacking these highly conserved residues, they maintain the Ig fold, and possibly through an increase of the hydrophobic contacts. So another example showing the core interactions. So you can see the tryptophan here, the disulfide bond here, and also the anchor of the E strand, which is important for the stabilization of the tryptophan. So here I show an overlap of IgV on the right, and then the other types on the left, which include the C1, C2, ISET, and fibronectin. And you can see that the orange has a very good overlap. And in the bottom, I show how they are in contact through backbone interactions with one another, although they actually also form side chain interactions, but we are not showing that for clarity only. So we also analyzed the stabilization of these contacts. So we perform distance analysis for each of the atom-atom contacts within each strand, between each strand and another. So you can see that it's highly stable. It remains stable throughout our 100 nanosecond simulations. And this is another example where we apply our number scheme, and you can see how we can locate both the conservation and the symmetry as well. So this in the bottom here is one example where we can show both the conservation of these residues in both types of and both monomers. So this is the CD8 alpha and the CD8 beta forming this dimer, and we can see that this residue orients kind of symmetrically with respect to each other, even though despite being the same residue. So whereas this is another case where it's actually a different strand. So this is the C strand and this is the C prime strand, but so they are kind of not conserved in the residue position, but they are conserved in the 4D quaternary symmetry and the 3D the quaternary symmetry. And here on the right, you see an overlap of both the VHBl dimer and the CD8 alpha beta dimer. And you'll see that there is this important interaction that is separating the two chains. And in the case of VHBl, it's actually the same residue and hold up the same place. Whereas in the CD8 alpha, they do hold up the same place within with respect to the chain, but they are different but similar residues. So you can see that here in the VHBl, actually they are oriented very perfectly with one another, and the axis of symmetry of the two chains actually goes through these two contacts. Another example is the CD19. CD19 is a complicated case because it has a big domain. It does not seem like an Ig domain at first because it's much bigger. However, through overlap of the domains, both in the sequence and in the structure, we notice high similarity between the two domains of the CD19. And so to give you an understanding, so in CD19, it goes from A, B, C, C prime of one domain, and then it continues that with the other domain. So then it goes on to finish the other domain. It comes back to finish the first domain right here. So you can follow the arrows. So there is 4 protodomains. The protodomain of the first one, and then the protodomain of the other, and then backwards. And here I also have an alignment both of the sequence and the structure orientations of the CD19. So this is essentially the sequence, and you can see that it starts with the blue domain, and then at some place it changes to the red domain, continues the red, and then goes back to form the blue one like we showed at the top. But here we have the structure-based alignment. And you see that they are highly conserved, and you can see that they have the distinct they have the typical Ig characteristics, such as the presence of the disulfide bond between B and F, but also the presence of tryptophan on the strand C. And you can also see here the helix conservation between the two domains. This is a small helix that is present in the same position with respect to one another. For the future direction, we aim to apply our number scheme not just to the strands but also to the loops like CDR3. We also aim to apply our number scheme to the other Ig-like domains, which are more complicated cases like CD2, CD3, CD19, major histocompatibility complex, microglobulins and TCR, but also neural receptors like Ty1, NCAM, and Sidekick, and other proteins that do look like Ig domains but have not yet been classified as Ig domains due to the complex folds like p53. We also aim for the rational design of therapeutic nanobodies and antibodies that specifically help target cancer cells, as well as for targeting of membrane receptors in order to stabilize and crystallize them. And that can be done through rational modification of the Ig domain, which would be facilitated by our number scheme. And you can see here in the bottom structures of the ones we would like to analyze next, which we actually have already started to. So this work has been possible with the help of Tom, Jiao, James, which was supported by the National Center of Biotechnology Information of the National Library of Medicine at the National Institute of Health. I would also like to give special thanks to Philippe for his amazing work in this paper, which I recommend that you should read. This is actually the basis for our numbering scheme. All right, thank you very much for listening to my talk, and I hope you have a wonderful day.